Cardiac muscle fibers are striated, branched, and have intercalated discs. These are the red squiggly lines that you see here. The heart muscle must contract at the same time. They must be synchronous. So the intercalated disc has three features that helps with the synchronicity function of the heart. The first feature are the interdigitating folds that you see here. These pink and red folds are so close together that they increase the surface area of contact between neighboring cardiac myocytes. The second feature are tight junctions. They are also known as desmosomes. Desmosomes are these lavender structures that you see here. They're mechanical junctions that keep the myocytes from tearing away from each other when the heart has to contract in a forceful manner. The third structure are the green structures that you see here, and those are the gap junctions. They are ion channels that allow the free movement of ions between the cardiac myocytes. All these structures together help in the synchronicity aspect of the heart function. So we have interdigitating folds that increase contact, desmosomes that prevent tearing of myocytes, and gap junctions that increase the speed of communication between the cardiac myocytes. Let's compare cardiac muscle with the skeletal muscle. We just saw that cardiac muscles have intercalated discs to help with the synchronicity. Skeletal muscles do not have intercalated discs. The cardiac muscle uses a very dependable form of fuel, namely fatty acids. The body does not run out of fatty acids, so it makes the best kind of fuel for the heart. The skeletal muscles, however, use glucose as their primary fuel, which can run out. The body does run out of glucose within 24 hours. The secondary fuel for skeletal muscles is fatty acids. So when it runs out of glucose, it then uses fatty acids. But the heart cannot afford to do that. It always uses fatty acids. The heart muscle also cannot afford to fatigue. So it gets its ATP purely from aerobic respiration. Aerobic respiration produces large amounts of ATP and has no accumulation of lactic acid. The skeletal muscle, on the other hand, can go into fatigue because it gets its ATP from both aerobic and anaerobic respiration. Anaerobic respiration produces less ATP and results in also lactic acid accumulation. The cardiac muscle also cannot afford to go into tetanus. It cannot cramp. The reason is that it has a very long absolute refractory period, which is about 250 milliseconds. This allows the heart muscle to contract and relax. It has enough time to contract and complete its relaxation before a new contraction begins, so there is no tetanus. The skeletal muscle, on the other hand, has a very short absolute refractory period in its action potential, as a result of which it can go into tetanus. Your skeletal muscles do cramp. We can't have that happening with the heart muscle. If it cramps, it stops. Let's compare the shape of the action potential in a cardiac muscle fiber and in a skeletal muscle fiber. In a cardiac muscle fiber, the depolarization phase occurs where it goes up, it stays up for a little bit, and then comes down. In a skeletal muscle fiber, the action potential goes up and then it goes down. So in the cardiac muscle fiber, shown by the dotted red line, the absolute refractory period is very long. And in the skeletal muscle fiber, the absolute refractory period is much shorter. Now let's see how the action potential is generated in a single cardiac myocyte. We call it a non-nodal action potential to differentiate it from the nodal action potential that we saw earlier. Here is the graph. Time on the x-axis, time in seconds, and membrane voltage in millivolts on the y-axis. Let's make a few markings. Minus 90 millivolts, plus 20 millivolts, and 0 millivolts. At minus 90 millivolts, sodium channels open and sodium rushes in, causing depolarization and opening of many more sodium channels in a positive feedback loop, and the voltage reaches plus 20 very quickly. This voltage is maintained sort of by the opening of calcium channels and the slow inflow of calcium. The repolarization is brought about by the opening of potassium channels and the fast outflow of potassium. So we have it here, the depolarization, fast sodium inflow, the maintenance of the depolarization by slow calcium inflow, and the repolarization by fast potassium outflow. The maintenance of the depolarization is called the plateau, and as you can see, it is brought about by slow calcium inflow. This is what makes the absolute refractory period extremely long, as a result of which the cardiac muscle does not go into tetanus. Putting a pet down to sleep can be a very traumatic experience. This is accomplished by giving the animal a potassium chloride injection, which results in cardiac arrest and death. Let's see how a potassium chloride injection can lead to cardiac arrest based on what you know about a non-nodal action potential. So when a potassium chloride injection is given, the levels of potassium in blood increases because that's where it ends up. The potassium ends up in the blood. So the potassium levels in the extracellular fluid, ECF, is greater than potassium in the intracellular fluid, or ICF. This is the opposite of what it is normally, because we all know that there is much more potassium inside the cells than outside. Therefore, as a result of the potassium chloride injection, the potassium gradient is completely disrupted. So when the action potential happens and potassium channels open, there is no potassium outflow, or there is very little potassium outflow. If there is no potassium outflow, then there is no repolarization of the cardiac myocyte. If repolarization does not occur, it will not be followed by relaxation of the cardiac myocyte. So relaxation of the muscle also does not happen, and the heart goes into cardiac arrest, which results in death.